Ever wondered why comparability protocols are so crucial in biologics development? Let's dive right into it. Mastering biologics development. Here's a quick guide to creating effective comparability protocols with your partners at Incrisi. Comparability protocols, also known as CPs, play a pivotal role in the realm of biologics development. They are regulatory mechanisms used to assess and demonstrate that changes in the manufacturing process of a biologic do not adversely impact its quality, safety, and efficacy. Imagine, if you will, a manufacturer planning to tweak the manufacturing process of an already licensed biologic. Having a detailed plan for assessing the effect of this proposed change is where a CP steps in. The primary aim of these protocols is to ensure that both the pre-manufacturing and post-manufacturing products are comparable in terms of quality, safety, and efficacy. In essence, comparability protocols are like safety nets, ensuring that no significant alterations occur that would affect the biologic's intended use. Without these protocols, any slight alteration in the manufacturing process could significantly affect the biologic's intended use. Now that you understand the importance of comparability protocols, how do you go about creating one? Let's dive into an 8-step process that will take you from preliminary assessment to documentation and reporting. Firstly, conduct a preliminary assessment. This involves an initial evaluation of the current manufacturing process, the proposed changes, and its complexity. It's about identifying potential risks and impacts on the biologic's quality. This sets the stage for the comparability study that's to come. Next, you'll move to risk-based planning. Here, you'll develop a plan categorizing the changes as either major or minor. Major changes call for a more extensive evaluation, possibly including both non-clinical and clinical assessments. Minor changes might only require analytical assessments. Then, it's time to implement your development studies. You'll systematically execute these studies, starting with analytical assessments and then moving on to further process development and, if needed, brief clinical studies. This methodical approach ensures a comprehensive evaluation at every stage. The 4th step involves analytical and functional testing. This is where you'll conduct thorough testing to assess the molecular structure and functional activity of the biologic. The aim is to confirm that the product maintains its intended characteristics post-changes. In step 5, you'll be evaluating and setting specifications. You'll review and update the product specifications and expiry dates to reflect the impact of the manufacturing changes. This is vital for maintaining the product's quality and regulatory compliance. Next, you'll conduct pilot studies and monitoring. This involves testing the new process in a controlled environment, and following successful pilot studies, you'll implement continuous monitoring to track the product's performance and ensure ongoing compliance. In the penultimate step, you'll move into process validation and regulatory compliance. You'll validate the updated manufacturing process and ensure adherence to all regulatory requirements. This step is crucial for ensuring the safety and efficacy of the biologic. Finally, you'll focus on documentation and reporting. This means maintaining detailed documentation throughout the process and preparing and submitting reports as required for regulatory review and approval. A CP can be submitted in an original application or can be submitted in a PS. A pass containing the CP must be approved by FDA before the proposed changes are implemented, as outlined in the protocol, and the product is distributed in accordance with the approved reporting category. A CP can be useful in providing predictability for the implementation of future changes to an approved product, including its manufacturing process. By delineating the specific approach to be used to evaluate one or more future changes and the rationale for that approach, you can gain FDA's approval of the plan well in advance of the need to implement the change. By following these steps, you can ensure that your biologic maintains its intended quality, safety, and efficacy throughout the development process. Let's recap what we've covered today on developing effective comparability protocols. These protocols are essential regulatory processes to ensure that any changes in the manufacturing process of a biologic don't negatively impact its quality, safety, or efficacy. The aim is to ensure that both pre-change and post-change products are comparable in their intended use. The process involves a series of steps: preliminary assessment, risk-based planning, implementing sequential studies, analytical and functional testing, setting specifications, process validation, pilot studies, and documentation. Each step is crucial in its own right, from identifying potential risks to validating the updated process and ensuring compliance with all regulatory requirements. Maintaining detailed documentation throughout the process and preparing reports for regulatory review and approval are also vital parts of the process. This comprehensive framework provides a clear path to developing a comparability protocol for biologics. Remember, the goal of a comparability exercise is to ensure that the pre- and post-change drug product is comparable in terms of quality. With this guide, you're now equipped to develop a robust comparability protocol for your biologic. Efficacy, which means approval in Greek, was founded on the premise that biotechnology, medical devices, and pharmaceuticals hold the promise of a better future for everyone. Making good on that promise requires innovative approaches, world-class science, regulatory know-how, and a team dedicated to bringing all of it together. We're more than a team of experts. We're your partners,